Okay, let's start. Good morning, afternoon, and evening. And hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요. And hello all. Welcome and thank you, SynBYSS people, for joining us the 182nd seminar, and I call it on NIBIB and NIH Day. As the holiday season is approaching, I think more of my loved one, family, and friends who live far away from me in San Diego. So I think those people more than ever. If you cannot visit or meet them in near future, or regardless of our plan for the holiday, why don't we just call them and say hello and say I love you? Or just simply texting them. I think giving is the foundation of changes, and the best gift is attention or love, not expensive diamond or something. Okay, it is my tremendous honor to introduce very briefly our pioneer speaker, Dr. Robert Berry. He is a program director interested in magnetic resonance imaging at National Institute of Biomedical Imaging and Bioengineering, or in short, NIBIB, the National Institute of Health. I believe I met him last year at NIH, where NIBAB hosted the annual synthetic biology meeting, and I gave my talk over there. At that time, he kindly accepted my invite to give a talk at SynBYSS. I especially thank NIH for their support because a total of 8 program directors, the largest number of pioneer speakers along with the NSF, participate in this webinar series. This is a 17% among total of 47 funding agency directors who join us as pioneer speakers. As I mentioned earlier, giving is the foundation of change, and I believe those funding agency directors and all the speakers in the seminar series giving their time to change entire world in a better place, I believe. Robert, thanks so much for your support to the research community, shaping the field, and also the virtual podium is also yours now. And thank you again for your time today.
Well, thank you very much, Taesuk, for the very kind introduction. It's a pleasure meeting you, Rebecca, here this morning. Yeah, I just wanted to talk for a few minutes about how I got to where I am right now. And I think it's kind of an interesting story because, when I started my career, I guess you could say the origins were back in, grade 10, 11, and 12 as I was finishing up high school. The funny thing is I hated biology. I wasn't good in biology. I didn't want to have anything to do with biology, but I liked solving complicated problems. Now, that's a bit of a foreshadowing of where we're going for this because it turns out that some of the most difficult problems in the world are biological, but I didn't know that at the time. So I was interested in computers. I loved computers. I loved programming. So I thought I'm going to, pick the hardest program when I got my bachelor's in computer engineering. And then I thought, well, this is pretty fun. I'm going to keep going. And then so I decided to pursue a master's in computer engineering, focusing on like basically telecommunications and signal processing. Um, and while I was doing my master's, I remember actually I think I was grading calculus papers on a Friday night. I saw an advertisement for looking for volunteers for brain studies. Basically, it was recruiting for MRIs, and they were looking for healthy participants. And I thought, what the heck? I love MRIs. I've never had one, but it's cool technology and I don't know how they work. But, I'm happy to be like what you call like just a, chunk of meat in the scanner, donate my brain for an hour and get, $50 or whatever it was. And so I did. And I loved it. I thoroughly enjoyed being a participant. And I guess I gave them good data because they kept calling me back. And, I participated in several MRI studies while I was finishing my master's. And then I thought this was like really cool. You can use an MRI, and actually, it wasn't just strict MRI, it was actually functional MRI. So I was participating in studies where you're using the MRI to try to learn something about how someone's brain worked. And I thought, this is so cool. I want to do a PhD, in functional MRI. And that's what I ended up doing. So, when I was teaching my students, I said, you don't know where people are going to come from in their journeys. And when you deal with participants and patients, some of those people, might get excited about the technology that you're investigating. So I went and I did a PhD in high field MRI. At the time in Canada, it was 4 Tesla in London, Ontario. It was the highest field for human imaging at the time. And I was there for about 4.5 years. And then after that, I wanted to do even more. And so I thought, well, I want to get the cutting edge of technology. And basically, I was imaging the brain. That's what I was doing kind of every day, and I was analyzing data. And going back to my earlier comment, I mean, I had to learn and catch up a lot with a lot of biology because I got grade 9 bio, basically. So I had to teach myself a lot of biology and trying to get quickly caught up. But it turns out that all the techniques that I had learned, this is kind of the interesting segue, were not wasted. I was learning signal processing, fractal analysis, multidimensional analysis, neural networks, et cetera, et cetera, et cetera. I remember back in the, late 90s when AI was, being talked about almost every day. And then it kind of quieted down for 2 decades. And here we are again. And all people are talking about is AI. So, I mean, this is now like the second or third, AI revolution, if you go back far enough. And so all the techniques that I had learned actually were applied every day. But instead of taking a look at telecommunications traffic, I was looking at brain traffic, traffic transmitting between nodes in a brain, hemispheres, lobes, going from one piece of cortex to the other, and the highways between were the white matter. So basically, everything that I learned was applied to, new studies. From then, I did my postdoc fellowship at Vanderbilt at 7 Tesla, so even going to higher magnetic fields for human imaging. At the time, I think there was maybe only a couple dozen 7 Teslas. And at a higher magnetic field, much higher than you typically see in a clinical setting, you can learn a lot more about how the brain works, but it is very technically challenging. And that's once again how my background in computer and biomedical engineering was leveraged because I was basically developing new techniques and new hardware and new software and new algorithms, and we were applying them to learn more about the brain. And then from then, I wrote a couple grants to the NIH, and I got funded. And I was recruited up to the Massachusetts General Hospital in Boston, and I had appointments at Harvard Medical School and at MIT. And I was there for about 8.5 years, and I ran a research lab. And the research was basically continuing once again the last 15 or 20 years of my career at that point. We're looking at the development of new biomedical signal technologies, analyzing data, acquiring the data also not only from the brain but also the human spinal cord. It was a very technically challenging and understudied area. And so we did that for close to a decade. And then I saw a position open up at the NIH for this program director position. And in this position now that I'm currently in, I'm helping to sort of like, guide, the next hopefully 10 to 20 years of, MRI research in the field that is related to, our institute, NIBIB. But yeah, so that's just kind of a little bit of a background. And I like to just tell people that are, watching this that, I remember my dad used to say, like, life is what happens when you're making other plans. Like, never in a million years would I have guessed that I'd be studying the brain when I was in high school because that was biology and biology was like icky and hard and I didn't understand it. Never in a million years did I think I'd be doing, like a PhD when I was doing my bachelor's. That never crossed my mind until I got excited about research. At the time I was living in Canada, never did I think that I'd go down to the US. Well, now I've been in the US for almost to 20 years now. So, never did I think I'd go up to Boston and spend time at MGH and Harvard and MIT, and I was there for 8 years. And never did I think that I would, now be employed by, the federal government. So basically, if you had to take a look at my career, everything that I never would have imagined is exactly what happened. It's one of those things like a probability curve. Like the probability of any one thing is like zero, but happens all the time. Yeah, I just want to tell people, never close doors. Kind of always, keep your mind open. I also tell people it's important to make connections. Through this seminar series, we're reaching out to people around the world in different time zones. It's important to get to know people. It's important to like, understand that all of our stuff is connected. If Rebecca and I have a virtual coffee together, I'm sure we can find quite a few areas where her interests overlap with my interests, even though I have known nothing about what she does. I think that's one of the great things about research is, like we're all part of this web, we're all connected in some way. And yeah, it's been a fun career for me, and it's I never would have imagined that I would be here speaking today, but here I am. So thank you very much for the meeting with me, in person, and kindly inviting me to be a part of today's talk.
Oh my goodness. You know what? That was one of the most exciting, inspiring talks in recent, month or recent years. Thank you so much.
Thank
I'm
you.
100% agree with you. Starting with, I hate biology in high school because my high school teacher in biology is horrible. So I just choose the chemistry or applied chemistry, but somehow I got into the organic chemistry and then I love it. And then I start to
Yeah.
take biochemistry and then, oh my God, biochemistry is so cool. And then microbiology, another department, molecular biology. And then I am here now doing everyday microbiology or whatever the biology. So that is a fantastic because I'm getting a lot of questions from young people whenever I'm, meeting those people. They ask, very serious question. They worry about which choice I need to make. And then also I sometimes kind of giving them advice like, that's my question typically in the past, but this is the wrong question I'm asking. That was the what's next. That's my typical question in the past, but I realize it's a horrible question because they're giving a lot of pressure to those young people.
Yeah.
When I changed recently my question from what's next to what makes you happy.
That's right.
And then you
That's right.
found the way to go because you just follow your gut feeling and then you got excited and then you just kind of end up this amazing position because you're just excited and you just follow your dream or even just small incident or small opportunity, but that become the big, interesting new pathway. And because of life is not linear, this is complex, non-linear, path, and then that's why how you end up in at NIH. So this is amazing example. I want to watch this one again myself, or sending that to my daughter because my daughter constantly worrying about career in college.
You're absolutely correct. You're absolutely right. Whenever I would have a, bad day or a bad week with whatever I was doing, like there's ups and downs like everything in life, I would try to ask myself the question like, am I happy? In general, yes. What else could I do? And I think about other potential opportunities, and then I would say like, would those things make me happier than what I'm doing? And the answer was basically always no. I could not think of something else that had the potential, as best as I could, opine, to make me happier. And so that was really one of the guiding forces, and I really do think there's not one only path, there's no, this is the only thing I can do with my life. I think there's probably many choices, and we just have to pick the best one given, data driven, you just said earlier, all the data available today, I will make a decision, based upon what I can do. But yeah, exactly. That's the thing. I try to find, I always like to try to find a job that I like because then I never have to work a day in my life.
Absolutely. That's the beautifully, you said that. And I love it. So, oh my goodness, this is really inspiring. I need to, yeah. So I never thought I'm doing this kind of seminars I mentioned earlier, more than 4 years, but I'm doing so. So you never know. I'm in the future. So So young people just kind of dream high and then follow your gut feeling, and then you will find some opportunity, and then never give up. And that's kind of one thing I wanted to say. And then, yeah, wow,
Great.
I kind
Thank
of
you.
very impressed. Thank you. So I need to move on because even though I'm kind of very impressed, I have the job to
Of
do.
course.
All right. Thank you so much. It's
Thank you.
fabulous. I love it. All right. So now the main speaker of today with a longer introduction, Dr. Rebecca Wilkes was a postdoctoral researcher at the National Renewable Energy Laboratory, mentored by Dr. Greg Beckham and also Dr. Alison Warner, both of them actually the speaker of this series. And then suddenly or accidentally, I found she now uh, a synthetic biology staff scientist in the bioscience division at ORNR or Oak Ridge Laboratory, and also a joint faculty professor at the University of Tennessee as part of the UT Oak Ridge Innovation Institute's Circular Bioeconomy Systems Convergent Research Initiative. I just found that information just in time. Otherwise, I would be very embarrassed introducing in the wrong way. Congratulations again, the new position. She earned her MS and PhD from Cornell University under the guidance of Dr. Ludmila Aristide, where she was an awardee of the NSF Graduate Research Fellowship Program. Actually, Ludmila and I currently are preparing a paper on C13 tracing, and what a coincidence.
That's
During
wonderful.
her MS and PhD, she utilized a multi-omics approach with particular focus on metabolomics and 13C tracing to investigate carbon flux and regulation of native cellular pathways involved in the catabolism of natural and xenobiotic compound. Currently, she utilizes genetic engineering and systems biology approach to understand microbial metabolism and optimize microbial pathway for bioproduction. Rebecca, congratulations again. Thank you so much for your time today. Please take it away. All yours now. Thank you again.
Great. Thank you so much. Can you guys see my screen? Is that showing the
Yes.
slides? Wonderful. All right. Thank you, Dr. Moon, for that introduction and the invitation to be able to talk today. And also thank you, Robert, for those inspiring words. That was really great to hear about your career path more and making sure you're taking advantage of every opportunity as it comes to be. That's kind of how I feel going into my position at Oak Ridge National Laboratory. Today, I'm going to be talking about some of the work that I did as a postdoctoral researcher at the National Renewable Energy Laboratory, which was part of work in the Center for Bioenergy Innovation, which is one of 4 different bioenergy research centers in the US, and they all work towards converting process-advantaged plant biomass into biofuels and bioproducts that are relevant to industry. And I'll be continuing to do work within CBI in my new position as a staff scientist at the Oak Ridge National Laboratory. And just briefly, I wanted to say that Oak Ridge is in Oak Ridge, Tennessee. It's near the Great Smoky Mountains National Park, and it has this mission of research with impact. So delivering scientific discoveries and technical breakthroughs needed to realize solutions in energy and national security, provide economic benefit to the nation. I feel really fortunate that I've been able to work within the National Lab system because I think it offers this bridge where you get to work with researchers across academia and industry, as well as other national labs and federal agencies. And it's also kind of uniquely situated so that you can kind of do these long-term sustained R&D projects and kind of fill that gap between more of the fundamental research that you see in the academic sector and more applied research that is needed for the industrial sector. As Taesuk was saying, I work within the Biosciences division at Oak Ridge National Laboratory, and one of our strategic goals is working towards engineering a circular bioeconomy. So when we think of a circular bioeconomy, we can think of this idea that nothing is wasted. We're really optimizing and taking advantage of every carbon atom and to improve efficiency within this kind of closed system. For instance, when we're thinking about using plant biomass, we want to take advantage of every fraction of that plant biomass, whether that's the cellulosic fraction or the hemicellulose or the lignin fraction. We want to see how we can convert that into something valuable, something like materials or products. Additionally, when we're thinking about these kind of new materials, we want to think of the end of life. So we want to think of how we can make them recyclable by design, or how we can upcycle them into new products or new materials to keep the carbon moving within this kind of closed loop. So the mission here is to be able to design these biological systems for efficient production of bioenergy, bioproducts, and biomaterials, which is a big passion of mine moving forward with my research. In order for a bioeconomy to become established, we really need these integrated solutions and these multidisciplinary teams. So as a synthetic biologist, I work a lot with microbes, but I also work a lot with chemists and engineers and economists and things like that, so we can understand how we can take these complex, heterogeneous feedstocks, things like plant biomass or mixed plastics or waste streams, convert them into these complex streams that then our biology can come in, where we can harness the bacterial metabolism and convert these mixtures into a single product of interest, whether that be a new polymer like polyhydroxyalkanates or other products like diacids or pyridines or aromatics that can be made into these new polymers. The work I'm going to be talking about today is on lignin bioconversion. So lignin is this kind of complex aromatic polymer that can make up to about 40% of plant biomass and can be deconstructed chemically into these really heterogeneous mixtures of aromatic and aliphatic compounds. They can be free monomers, they can be really complex oligomers and polymer components. And the goal is that we take that mixture, feed it to a microbe of choice. That makes one product. It can be separated out and then polymerized into either direct replacement chemicals or polymers like plastic bottles, or ideally into some of these newer performance advantaged polymers as well. The organism of choice for this work has been Pseudomonas putida, which is a really exciting organism because it has this kind of broad catabolic capabilities to utilize a lot of different aromatic compounds. It has this really great stress tolerance. It's been cited for its use as a cell factory to make a number of different bioproducts. And there's a lot of genetic tools available to continually manipulate its metabolism into whatever products or other tolerance mechanisms that we need. However, when we're thinking about using a host for something like lignin bioconversion, it's really important that we constantly reevaluate our hosts and our choices and compare our microbes to new microbes that are coming out and it being discovered that have these potential new capabilities or processes that are really interesting. So some of the work that I started by doing was comparing Pseudomonas putida to some other putative host strains for lignin bioconversion. So in this work, I looked at KT2440 versus 6 other different strains that have been cited in the literature for ability to use some of the more complex lignin linkages, as well as having some tolerance and capabilities on aromatic compounds. And to start, we screened the strains on increasing concentrations of a mixture that's relevant to a lignin strain. So in this case, I used a mock alkaline pretreated liqueur from Cornstover. So it has a number of aromatic and aliphatic components in it, but these are model compounds. And then I also screened across single compounds as well. So p-coumarate, ferrylate, and acetate are 3 of the compounds that are the highest abundance in these strains. And one of the things we looked at is their growth across the increasing concentrations and as well as their rate. And you might notice here that there's this nice bar that goes all the way across and that indicates that KT-2440 was able to tolerate even the highest concentration that it was tested. And also it's a little bit darker green here, which shows that it has this really fast maximum growth rate. So again, we're showing that KT-2440 has a rapid growth and high tolerance to some of the compounds that are commonly found in these lignin streams. This work was a nice collaboration within the Center for Bioenergy Innovation, as well as two different bioenergy research centers, so the Great Lakes one and the J-Bay. Not only do we want to look at just those model compounds, we wanted to look at an actual lignin stream as well. So I fed all the strains 25% alkaline treated liqueur from corn stover and looked at the amount of lignin content that decreased after 120 hours. And with the exception of PD630, which didn't go very well on this stream, we saw about 10 to 12% of the lignin content decreasing after those 120 hours. And this content that decreased is pretty comparable to the amount of aromatics that was in the stream that were kind of freely bioavailable, things like p-coumarate that were released from the alkaline pretreatment. So it didn't necessarily confirm that there was any of the oligomeric components that were being broken down. So to look at whether or not any of the strains were able to act on lignin linkages, we used NMR with the help of John Ralph at the Great Lakes Bioenergy Research Center. And what he found is that only the two Sphingolian strains had any appreciable depletion of any of the common linkages that we see in our lignin. And so even though some of these have been cited a lot for their ability to use the dimers, when we're looking at a really complex stream where it's more oligomeric polymeric component, we don't have a ton of that more complex components that are being broken down. And so that's where we kind of have to turn back to our chemists and be like, hey, we need new solutions to increase the bioavailable or easily and freely available aromatic compounds so that we can convert more of that towards our products of interest and have our cells utilize that carbon rapidly. Now, this was all done on the wild type strains and not any engineered strains. And we were talking about how we want to make bioproducts. So how do we actually use Pseudomonas putida as this host for biochemical production? And one of the ways that we do this is to look at how we can partition its aromatic and sugar metabolism to optimize biochemical production. What do I mean by that? So we have lignin, which is this complex aromatic mixture, but we're going to actually just focus in on one model compound here. So this is 4-hydroxybenzoate. And the goal is that 100% of 4-hydroxybenzoate is going to our product of interest. In this case, it's mucinate, which is an intermediate in one of the aromatic catabolic pathways. And it's a really cool compound because it's considered bioprivileged, where it can act as a drop-in direct replacement for dipic acid in some nylon polymers, or it can actually be used directly as a precursor and be used for performance-advantaged polymers as well. Now, if we have 100% of our aromatic going to our product of interest, that means we need to supplement with something else in order for the cell to grow and survive. So we can use glucose, which comes from the cellulosic component of plants, and that 100% of that is now going to go to our cellular growth and energy. How do we engineer this? All right, so if we take our wild type strain, KT-2440, it takes 4-hydroxybenzoate and converts it protocatechuate and then through the orthocleavage pathway to our intermediates for biomass growth. When we want to engineer mucinate production, we have the strain CJ781, where PCAHG has been deleted so that we don't go through the orthocleavage pathway. We've added in a heterologous protein, ARY, to convert PCA to catechol, as well as adding ACDBD, which helps with the prenylated FMN cofactor. From there, we go back into the native metabolism, and catechol can be converted to mucinate. And here we've overexpressed CatA1 so that we can increase flux from catechol to mucinate. In the strain, we've also deleted CRC so that we don't have catabolite repression and we can have co-utilization of our aromatics and our sugars. We've also deleted CatBC so that mucinate doesn't go towards biomass. More recently, Eugene Kucha found that if you replaced POP-A, which can only use NADPH, with PRA-I, which can use NADPH or NADH, we can improve our cofactor ratio NADPH so that we have a higher pool of it. And NADPH is important not just for biomass growth, but for a lot of stress responses as well. And they found that that ended up improving mucinate production as well. So now we have a strain that can make mucinate secreted out, and that can be upgraded into performance advantage products. And this strain actually performs fairly well. So when it was grown in bioreactors, fed 9 millimole per hour of peak fumarate, they can get titers around 39 grams per liter with yields around 100%. The limitation really here for industrially relevant conditions is the productivity. So they're only getting about 0.5 to grams per liter per hour of mucinate being produced. So that's one of the limitations for the strain, and we want to be able to pursue that. But before we kind of dug into how we can make mucinate production faster, we really wanted to reassess the strain health, because we have these heterologous proteins that are being constitutively overexpressed in this organism, and as well as a number of overexpressed proteins, and that can end up causing stress to the organism. So we wanted to take a step back and be like, okay, how is glucose metabolism being impacted by our engineering of mucinate production? To do this, we turn towards omics analysis, which is dear to my heart. In this case, we used proteomics, which was done by Rich at Oak Ridge National Laboratory. And we did 13C metabolic flux analysis, which Patrick Southers assisted us with at Penn State. And there's two main findings here for our engineered strain. The first one is that it had an increased glucose uptake rate compared to our wild type, and higher periplasmic flux towards the oxidative product 2-ketogluconate. So Pseudomonas has this really cool metabolism where glucose comes into the periplasm and can be oxidized to gluconate or to 2-ketogluconate. And this puts electrons onto ubiquinol, which can then release into the electron transport chain and allows it to produce some energy fairly rapidly from glucose as a substrate. Another finding we had from this is that the TCA cycle flux was higher in our CJ781 compared to our wild type strain, with a little bit of disagreement in terms of the proteomics in terms of for malate dehydrogenase, but in general, on the oxidative side of the TCA cycle, which makes our NADH, we had this higher flux that agreed with our protein data. So that's ultimately going to have impacts on the energy state of the cell. And one of my favorite analyses to do is to look at our ATP supply compared to our ATP demand when we're using these 13C metabolic flux analysis. So we can look at our supply from catabolism and oxidative phosphorylation compared to our demand from anabolism or catabolism reactions as well. In our wild type strain, they're fairly balanced. However, when we look at CJ781, we have this kind of apparent excess in ATP that is produced compared to what the demands are that are in the model. So our model is not all inclusive. There are other reactions that are not included. So we could kind of look a little bit deeper into our proteomics data to see if there's any stress responses going on. And in particular, I was interested to see what was happening with some of the enzymes that consume ATP. What we find is that looking at these ATP consuming reactions or enzymes, CJ781 had significantly more enzymes that were higher expressed that consumed ATP compared to KT2440. So these could potentially be taking up some of this apparent excess in ATP production. But we also wanted to measure the ATP that was in the cell to look to see if there were actual differences in that total ATP pool between our engineered and our wild type. And what we found is that CJ781 actually had threefold more ATP than KT2440 in terms of its pool. So not only are we having this increase in potentially ATP consuming reactions, we also have this surplus of ATP in our engineered strain. And it's a way that it can potentially be dealing with the stress from mucinate production as well as heterologous protein production. We wanted to dig into additional potential stress responses that are happening, and proteomics is really helpful for finding some potential stress responses. So by looking into the proteomics, we found that acetate permease, which can either export or uptake acetate and pyruvate, it was really high for our engineered strain when it was grown on glucose and 4-hydroxybenzoate. And so we wanted to take a step and go, okay, so what is happening extracellularly? Do we actually have secretions of pyruvate and acetate? And the answer is yes, we did. we ended up having pyruvate and acetates accrued at around 2.1 and 1.8% of our glucose uptake, which you might say, okay, that's pretty small, but it's still really important to figure out that there are these kind of bottlenecks that are happening in central carbon metabolism. And additionally, when we're thinking about downstream separations of our mucinate, which is a diacid, we don't necessarily want secretions of other acids that are going to potentially make that separation a lot harder. So one way we can look at how we can potentially mitigate this is, since pyruvate was building up a lot intracellularly as well, we overexpressed GLTA, which is a commitment step into the TCA cycle. And by doing that, we ended up having a 4.2-fold decrease in our pyruvate secretion and a 1.7-fold decrease in our acetate secretion. So we can kind of mitigate these bottlenecks in the central carbon metabolism and decrease these secretions. So all of this was looking more at how glucose was metabolized and that energy. And we found that in general, Pseudomonas fetida is pretty robust. It didn't end up impacting its growth rate. It's still growing well. And it has these strategies to mitigate that stress from mucinate and heterologous protein production. So now we want to go back to that question of how can we improve mucinate production further? How can we improve the rate that mucinate is produced? So let's go back to that aromatic catabolic pathway, which has 3 steps here. And we look at exponential growth, and we have a mucinate secretion rate here that's happening, but we also have secretion of catechol and protocatechuate, the 2 intermediates in the pathway. And if we look at that relative to our 4-hydroxybenzoate uptake rate, protocatechuate is secreted at 26% of that uptake, catechol is secreted at 17% of that uptake, and so that's leaving only 57% of that uptake rate to mucinate production. And so those are pretty significant bottlenecks in this pathway that we want to try to mitigate. And when we think about bottlenecks, we always want to try to eliminate a downstream bottleneck before approaching an upstream bottleneck. So to start, we wanted to overexpress CAT-A2. And so by overexpressing CAT-A2, we were able to eliminate our catechol secretion, decrease our protocatechuate secretion 1.5-fold, and as well increase our mucinate production rate by 3-fold. So now when we look at it relative to our 4-hydroxybenzoate uptake rate, we have only 10% secreted as protocatechuate, leaving 90% of it going to mucinate. But we still have 10% secreted as protocatechuate, so we want to now try to engineer that further. And the most effective strategy was actually altering their ribosome binding site in front of AROY to improve our translation of the enzyme. So in this strain, we were able to decrease our protocatechuate secretion 2.8-fold, as well as increase our mucinate production rate. So now during exponential of our 4-hydroxybenzoate uptake rate, 4% of it's going to protocatechuate, leaving 96% of it to be mucinate secretion. So this is really indicating that our productivity is going to be substantially higher in these strains compared to our original strain, CJ781. But all of these were done in shake flasks in batch growth. So it's only about 25 mils. So we really want to assess these strains. We have to go back to our fed batch bioreactors to see if they do actually outcompete our original strain, CJ781. So feeding these strains in fed batch bioreactors at 10 millimole per hour, we do see that both of these strains are substantially superior to our original strain, CJ781, when fed that 4-hydroxybenzoate. And in fact, at this rate, CJ781 ended up dying off after about 48 hours and having substantial accumulation of catechol, as well as protocatechuate and 4-hydroxybenzoate, which kind of killed it off rather rapidly. Whereas both the other engineered strains that we tested were able to grow pretty comparably for 72 hours, and the RW133 strain ended up with a titer of 46 grams per liter at a rate of 0.9 grams per liter per hour and 100% molar yields. So this is a huge improvement in terms of our rate coming from 4-hydroxybenzoate. But can we push this even further? So then we tried a little bit higher feeding rate, so 13 millimole per hour. And again, we have no catechol accumulation, but we ended up having a little bit of accumulation of our substrate, 4-hydroxybenzoate here, which meant that the strains didn't perform quite as well in terms of titers. So our titers were around 36 grams per liter. but we did end up having a higher rate of production. So our rate was 1.1 grams per liter per hour, which is pretty awesome for these strains, and a little bit more of a step towards what our industrially relevant processes need to be. All of this was for mucinate, but we've talked about a lot of potential for different biochemicals and bioproducts. So what about some other biochemicals of interest? So let's talk a little bit about beta-ketoadipate. Beta-ketoadipate is another diacid. It's also in the aromatic catabolic pathway with potential to be made into nylon products with performance advantages. It has similar engineering to our mucinate production strain, where we have Pro-I replaced, we have CRC, or Pro-I added instead of POP-A, we have CRC knocked out, we have VAN-AB overexpressed to help with G-type lignin monomers going into protocatechuate. And then beta-ketoadipate is in the orthocleavage pathway, so we don't need any heterologous rerouting into catechol. And then here we've also deleted PCA-IJ and LVAE to prevent beta-ketoadipate going towards growth. And this work was done by Ali Werner, who is one of my advisors at NREL as well. And this strain works very well on different model compounds. So on p-coumarate or ferrolate, which are model compounds for lignin, as well as a ratio of the two, and that alkaline pretreated liqueur extract that I was talking about when we were comparing these different host strains. And what we see is that you can get yields of around 100%, especially at 18 millimole per hour. And we can get productivities over 1 gram per liter per hour and titers approaching 40 grams. So this strain is performing really well, but one of the things that we like to do a lot at NREL and as well in anytime we're assessing a new product is to do some techno-economic analysis to look at the minimum selling price breakdown and figure out additional improvements that we can make to make this process even more cost effective. And so right now we're getting about $2 per kilogram as the minimum selling price for beta-ketoadipate, which is excellent, but we can constantly make this better. And one of the things that this highlighted is the cost of using glucose. So we can reduce the price of beta-ketoadipate or cost of making beta-ketoadipate by decreasing that ratio of glucose to the lignin-related components, or by potentially removing glucose and using something that's already in the lignin stream. So you may recall that I talked a little bit about acetate, and so acetate is common in different lignin streams. So we wanted to assess whether or not we can feed the cells acetate instead of glucose to support the cellular biomass growth, and then use coumarate and ferrolate as these model aromatic compounds for the lignin stream to make beta-ketoadipate. We assessed this in fed batch bioreactors, feeding the growth substrate, glucose or acetate, as well as the two aromatic compounds. And you'll see that the growth over time, while the acetate-fed cells ended up having a slower delayed growth, a longer lag, and a slower growth rate, they ended up with a similar final OD. And if we break this down into our phases of growth, so exponential or E phase, where we have the highest growth as well as production. And then we also have stationary, where cells are starting to die, and then decline phase when they are dying off. And if we look at our biomass yield here in terms of grams of biomass per mole of carbon consumed, whether that be glucose or acetate, we did see that the biomass yield was significantly lower in our acetate-fed cells compared to our glucose-fed cells. However, this is in terms of their biomass growth. What about their production? Did this impact their ability to make beta-ketoadipate? And no, it did not. So if we look at our beta-ketoadipate, we see that similar beta-ketoadipate production during exponential, and actually during these stationary and decline phases, we have a little bit higher beta-ketoadipate produced in our acetate-fed cells. And if we look at this in terms of rates, we saw similar rates and during exponential growth, but at stationary, we ended up having a significantly higher rate of beta-ketoadipate being produced in our acetate-fed compared to our glucose-fed cells. And that ultimately led to a higher titer for our acetate-fed condition compared to our glucose-fed condition, where we ended up with 42 grams per liter of beta-ketoadipate after 120 hours. So this is kind of highlighting that we could potentially switch out our growth substrate here and use acetate that's already present in the lignin streams to support our growth and our beta-ketoadipate production and reduce the cost of this process. An additional cool cost savings that we have is that by feeding acetate, we ended up having a lower requirement for base neutralization. So the acetate-fed cells, as they're consuming that acid, required less ammonium hydroxide to neutralize the diacid beta-ketoadipate. So we can not only save by not feeding glucose, we can potentially save by not needing as much base addition either. So this was one of the key questions in this project was, can we replace glucose with acetate? But another thing we really were interested in is what is happening, what are the metabolic responses that are occurring from that transition from exponential growth to the stationary phase and decline? Because that's really a question that can help us answer how we can potentially improve our bioprocesses to support a longer exponential and production phase. And just a brief highlight here, there are two stress responses that were really consistent across both growth substrates. So this is looking at our transcriptomics and proteomics data of our stationary or decline phase relative to our exponential phase. And superoxide stress in particular was over 1,000-fold increase at the protein level compared to exponential phase. So that's really highlighting that this was a huge stress response that is occurring later in growth. And then we also saw the start of iron scavenging, particularly in the RNA level, where the cell is starting to need a little bit more iron once we go in towards stationary decline phase. So these are two things that could potentially be mitigated by supplementing things like manganese to help with superoxide stress, as well as iron for those later stages of growth. So to summarize, I hope that this research is showing you some steps towards how we can use Pseudomonas putida to make new biochemicals and materials, potentially. I think it's a really cool host. I show again that it has this really high tolerance to aromatic and aliphatic acids that are common within lignin streams. It's also a very robust organism when it's coming to mucinate production. It does alter its glucose flux and increases ATP production, but this is a way for it to continue to grow at the same rate as the wild type strain and deal with those stressors from heterologous protein production and ATP production. And then we can continue to improve that strain in order to eliminate some metabolic bottlenecks that are in the aerobatic catabolic pathway, eliminate our catechol secretion, and increase our rates of production of mucinate. And then turning towards beta-ketoadipate, we can replace glucose with acetate or as a growth substrate so that we can reduce our cost for making the beta-ketoadipate product. And then we also see these stress responses in fed batch bioreactors, which were consistent across both growth substrates. So these are something that if we look at additional growth substrates, we might also see. So these are potential bioprocess improvements that we can make. And lastly, I really do want to acknowledge everyone that was involved in these projects. This is a very collaborative, these are very collaborative projects across national labs, bioenergy research centers, universities. In particular, I really want to thank Greg Beckham and Ali Werner, who were fantastic mentors to me during my postdoc, Rich, who did the proteomics, Patrick, who did the 13C metabolic flux analysis. So I thank you for your attention, and I'd love to open it up to any questions if there are any.
Fantastic talk. I'm already biased because I'm doing this type of work, but this is great talk. I will ask whether Robert's still here or whether he has some comment or question.
Yeah, I'm still here. I thought it was a great talk, and yeah, I don't have any questions. Like I said, I don't understand, a lot of biology. It's just just some beautiful work. And I just wanted to also comment that you did a phenomenal job presenting it. I think you're, one of those rare people that you can take complicated ideas, and do your best to make them, much more simpler and no more complicated than they need to be. So yeah, thank you very much for your presentation. It was wonderful.
Sure.
Appreciate
Thank
that.
you. I have too many questions, but I will probably only ask a couple of, during this official hour and then probably ask later. So the first simple question is, I used to be in the industry, so I always care about the techno-economic analysis, and then you showed beautiful data that's talking about $2 per kilogram. I believe you're talking about this is market price you're gonna sell, right?
Yes, minimum selling
So
price. Yeah.
so I know I'm in the gluconate. I used to also make the gluconate, but we haven't published anything because the reason was I couldn't find the right current market price of the not gluconate, the muconate, I'm sorry.
Oh,
I
okay.
couldn't find the current market price of the muconate. And then so do you know any how the current market price of the muconate? Because we know the Sigma Aldrich price, but that's not real
Mhm.
market price. And then I just want to know if that's high enough, I probably start to again the muconate production because this is the probably one of the reasonable product because all the lignin product all the way go to the muconate.
Mhm. Yeah, I don't know the exact amount off the top of my head. I know that we are working on some techno-economic analysis for it.
Mhm.
And I think instead of mucinate, you might even be able to find adipic acid instead
Yeah,
because it can be converted to that. And that's kind of more commonly used for
right.
polymers. So that would be probably the comparison you'd want to make is adding that extra step to convert mucinate to adipic acid, since that is kind of more standard right now,
So
rather than
acetic
using mucinate.
acid, if I remember correctly, this is only around $4 per kilogram each, right?
Yeah. So
So
that's
that
the
means,
thing
I
with
mean,
these.
yeah, that means the
Yeah.
$2 per kilogram, you have additional step. The still we need to improve, things. Otherwise, this is not commercially viable yet. Even though produce amazing amount. I think 40 gram per liter or something,
Yeah,
right?
40 grams per liter.
That's amazing. I mean, this is an amazing amount, but I still wondering whether including that everything and then making adipic acid that is economically viable.
Yeah, no, I agree. I think that's why we constantly want to find improvements and other strategies to really get industry to kind of latch onto this and take it to
Hmm.
the scale up, because
Hmm.
that's one of the issues right now is these are pretty small scales. So these bioreactors only go up to about 500 mils.
Hmm.
Uh, but we really need to be able to look at them at these higher, like more industrially feasible
Sure.
scales to see what we're getting in terms of our titers, rates, and yields.
Once you are successful, you probably retire earlier with $1 million in your pocket, I guess. And that might be, the interesting situation. But the second
Yeah.
question I have is, you also use the acetate. That's amazing. But have you done the new techno-economic analysis by replacing glucose with the acetate?
Not yet. So this work is still unpublished, and we're kind of figuring out the next steps to make it into a publication. But I do think that would be really interesting,
Right.
especially if you're looking at like alkaline pretreated liquor has acetate. It's actually the highest concentration in it in
Oh.
that stream. So you wouldn't be supplementing with acetate. You would pretty
Mm.
much just be feeding the lignin-related compound or that lignin stream and seeing how we can get production. And some of the other lignin streams as well have a decent amount of acetate, whether it's residual from the chemical breakdown,
Right.
or it is an actual breakdown product.
Okay, great. So I don't want to kind of ask all the question I have, but another question I want to ask, because you probably already know I'm heavily working on the rhodococcus
Yeah.
for more than a decade. And then the data you show is very interesting about the comparison between those things. To give some idea why I chose the rhodococcus over the Pseudomonas, when I start my position to 2012, I clearly see the Greg Beckham is leading the field. Also, there are multiple the people I admire, including Greg and also the Vito Del Lorenzo is doing those Pseudomonas. And then I look at this one, and then Pseudomonas is gram negative. And then to me, I'm done all the engineering before, and then to me, it's kind of not less challenging than Rhodococcus. So I kind of took I would call it the road less traveled This is one of the quote from the poem. I love that poem. And then I intentionally choose the other one because the other one, the rhodococos, also has a lot of great feature. Not necessary for that, lignin itself, but lignin breakdown compound, aromatic compound neutralization, high, lipid, production, and also very tolerant to that one. However, after 10 years of PD 630, I realized this is so painful because I need to develop all the genetic tool myself, including the transformation tool. That's painful, but I somehow kind of created Rhodococcus community. I all contribute to that one. So I'm okay with that one. But I'm moving a little bit more toward the JOS TI. I found Justia is much better than 630. So the question here is, you tested multiple different compound, not only the alkaline liquor, pretty liquor, but also the compound you chose. Is there any special reason you chose those compound? Also, is there any reason you didn't include the Justia but include the opacus?
Yeah, so for this presentation, I'm only showing 3 of the compounds that we tested,
Uh-huh.
but we also looked at vanillate, 4-hydroxybenzoate, syringate, vanillin, 4-hydroxybenzaldehyde,
Mhm.
acetate, lactate. So we did look at pretty much all the common ones that are in the stream for this
Mhm.
study. And
Mhm.
across everything, we really did see KT2440 having this really superior tolerance
Mhm.
to all of these compounds. And I would say vanillin and 4-hydroxybenzaldehyde in general had a lot lower tolerance across all the strains, which
Mhm.
are aldehydes, so you kind of expect that as well.
Okay. So in that case, I mean, the second question, so why not just TI, but the PD or Scopacus you chose for that study? Have you tried the just TI? Now I see the better with the just TI, and then I even
Yeah.
identify new bacterium that's close relative to the just TI, but a little bit different. And then that
Mhm.
bacteria is amazing, especially plastic upcycling. So now I switching from the okay, I'm dumping my 10 years old, research, with the PD630 and then moving into the Jostia more because the Jostia is better than the Opacus.
Yeah, so we didn't compare Just AI in this one because it was previously compared in some work by Davinia Salvatua
Uh-huh.
earlier, where she looked at, I think, more like 20 strains.
Uh-huh.
So this one was more of a close look at some of the new some of the emerging ones that were
I
of
see.
interest, especially things like Nova Sphingobia and Sphingobium, which have all the dimer and the different linkages in lignin. They've been kind of mapped out in those organisms. So we
Hmm.
wanted to kind of take a step back and kind of compare Pseudomonas again,
Hmm.
make sure that we're still seeing its usefulness on lignin streams.
Yeah, I agree with you. I mean, Pseudomonas, now I kind of understand. When I was studying, I make just go easier route with the more genetic tools. But somehow at that time, one idea is if I kind of use a Pseudomonas, can I get the funding? The answer was no. So I I chose the other way around. But
But I
now,
think it's really important that we don't just everyone work on one organism because
right,
they really do have different capabilities. So,
right,
like, you're not going to know the pathways or other things that you're going to map in until you've kind of studied some other organisms. So, a
right.
lot of the work I'm doing now is on a new organism, Comomus testosterone, that I didn't work in my PhD. And while it maybe won't be the eventual host for biochemical production, it has some really cool pathways that aren't in Pseudomonas. And
That's
so
the organism Ludmilla also using it, right?
yeah. So I did that in my PhD with her, and so now she's continuing to work with Comus testosterone and
Right.
find new interesting things that it can do. And so I think it's really important that researchers aren't all kind of studying the same organism because otherwise we're not going to continue to find these advances or realize that, oh, wait, actually, in this situation, this organism is better. So that was a big part of this paper as well. Yeah, Pseudomonas is really rapid growth we maybe would want to use something like rhodosporidium if we're going to be making lipids, because that's naturally produced in that organism. So
Absolutely.
yeah, and we might need to realize that it needs to improve tolerance under these conditions, but not under these conditions. So I do think it's important. I do make that caveat that, yeah, Pseudomonas is great, but we still really need to study these other organisms.
Right. So the one regret I have right now is at that time I'm not capable of dealing with the multiple organism, and then I wish I also start with the two organisms in parallel, Pseudomonas and Rhodococcus. That way I have better choices now. But I start to use, Pseudomonas several years ago. We published some paper, not necessarily metabolic engineering paper, but I also see the benefit of the Pseudomonas. And then I moving into more consortium, because consortium is the next thing, I believe strongly. And then, those consortium should have, those kind of amazing bacteria together. I call it the Avengers. Basically all the superhero together in one task, and then they doing amazing job, I believe. So it's a wonderful talk, and I now see the early over 11 Central Time. I have more questions, but I will ask that later. All right, so let's close. Uh, Thank you all for joining and staying today. We'll meet again on December 10, Wednesday, not the Thursday, again, 3:15 p.m. US Central, the same Zoom link. Because of the holidays, because of some other thing, we are basically meeting, almost, many days, 20 days later, 3 weeks later. And we'll have the Professor Pramod Angitka at IIT Bombay, India. I think the first dear Indian professor as a pioneer speaker, and also Dr. Burjita Ebert at University of Queensland, Australia. As usual, the follow-up informal chat will occur without recording. Please stay here if you're interested in chatting with us. I'll promote you to panelists who can speak and show your handsome or pretty faces if you wish. And I thank you again. I stop recording. Just 1 second.